Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there.
Hello, everybody. Welcome back to another episode of the A-Level Cookbook. Today, we're going to be doing the AQA A-Level Biology paper from June 2023. So now it says, give the three structural features found in all virus particles and describe the function of one of these features. So remember that viruses are different in the fact that they are not cells. So they're actually just nucleic acids like DNA or RNA surrounded by protein. They're not alive. So they don't respire. They don't, make their own proteins. They have to do it by using another cells. So the structures of viruses tend to be, so if I just draw a quick structure here, this is the core, the sort of like, the capsid, here. Within the capsid, you've got lots and lots of DNA, right? And that's that. But then how viruses work is that they infect the cell, take over the cell's machinery, and then start using that to replicate itself. So for example, it would get in and start mucking about with the cell's ribosomes to make more viral particles. The only way that it can get in is it needs to be able to bind to the cell's receptors and then get in that way. That's how a lot of viruses work. So what happens is you've also got these attachment proteins as well sticking out. What they'll do is they'll bind to a complementary receptor on a cell. This receptor might be for something else entirely. It just happens to bind to it as well, and then it can enter the cell like that. So here then, three structures include, well, the DNA or RNA, the capsid, and attachment proteins. So now it says to write the function of one of the named features. So here, a common mistake is that a lot of people describe structures that are only found in some viruses, like reverse transcriptase is only found in retroviruses like HIV, not every single one, or a lipid envelope. And other people describing things that you find in structures like that are bacterial structures, like, for example, capsules, plasmids. And other people were saying things like flagella and cell membrane. These are acellular, they're not cells. So the function of one named feature is its attachment proteins bind, so the attachment proteins bind to cell receptors, enabling entry. Here, many people did not say the word receptor or binding. They just kind of knew they have attachment proteins and they somehow mysteriously got in that way. The only way it can do that is by binding to complementary receptors and then getting in that way. Otherwise, it's not doing it. This has explained why viruses are described as acellular and non-living. So we need to compare what a cell does and what this non-living thing does. So if it's acellular, it lacks the same structure as what a cell would have. So they don't have cytoplasm, they don't have cell surface membrane, they don't have organelles as well, right? So immediately, one thing we can say is that they have no organelles. Alternatively, you could say that they have no cell surface membrane or they're not made of cells, right? Next, we need to think about what's happening in a cell. So in the nucleus, you've got your, DNA and, mRNA is made through transcription, it's translated to ribosomes, made proteins that get packaged in Golgi apparatus, put in vesicles, center, all of this stuff's going on, right? Viruses cannot do any of that, right? They have to, they can't do it on their own. They need another host cell to do it. So in that case, they have absolutely no metabolic reactions happening in them. They're non-living. Alternatively, you could say a specific one, like, so for example, they can't respire. So now it says, give one reason why antibiotics are not effective against viruses. If we think about how antibiotics work, is that they usually go for things within the bacteria or its structure. So, for example, its cell wall or enzymes within it. So this doesn't have any cell wall and it doesn't have the same bacterial structure. That's why antibiotics don't work. So they do not have a cell wall is what I'm going to go for. Here it says many students failed to understand what this question was driving at. The reason antibiotics are not effective against viruses was supposed to get the idea that viruses don't have the same structure as bacteria. That's what you were supposed to see from this question. Commonly, people were saying that viruses live/hide inside human host cells, so you couldn't get to them with antibiotics. That's more of like a GCSE level answer, and also not necessarily true, because you can have bacteria that live inside cells as well. Here, some people also then started talking about things to do with variability of antigens on the virus caused by mutations and talking about antibodies. Antibiotics are chemicals that go and destroy cell walls and things like that. They don't do immune system stuff. There is no immune system in this. Chitin is a polysaccharide. The chitin monomer is a beta-glucose molecule with one OH group replaced by an NHCOCH3 group. NHCOCH3 can be represented by this NAC. Fine. Figure 1 shows the monomer that forms chitin and the chitin polymer. Here's our monomer, and let's have a look where the polymerization bits happen. As you can see, there's an OH there and there's an O there. So clearly you can see there's a glycosidic bond forming there. It's a similar thing to, like they told you in the question as well, similar to beta-glucose. So that's what you'd expect beta-glucose to go, to look like. And the OH here was there, but it's been replaced by that. Fine. Now it says chitin has a similar structure to cellulose. Use figure 1 to describe 3 ways in which the structure of chitin is similar to the structure of cellulose. Well, if we look at cellulose, it's made of beta-glucoses that are joined together by glycosidic bonds, right? But the upper OH here then becomes lower on the other side. Can you see how it flips? It's the same thing here. So one of them already is that the monomers are flipped upside down. Now, if we think again about cellulose, what else is cellulose? So whenever you're thinking about bond making, bond breaking, and all that kind of stuff in biology, you need to talk about condensation and hydrolysis reactions if it's appropriate to. So, for example, if we're talking about proteins and carbohydrates and lipids, then you're going to be talking about that. Here, you also need to think about what type of bond is forming. So this is a polysaccharide. So it means there's going to be glycosidic bonds here. We can also see that there's those glycosidic bonds there too as well. So that's another similarity, is that it has glycosidic bonds. And if we think about our different sort of carbohydrates, starch, remember, can have like, it can be coiled as well as branched and straight, and unbranched. Whereas here, this is just unbranched. There's no branches here, and it's told you as well that it has a similar structure to cellulose. So if you can infer from this straight line that it's unbranched. Another similarity is that it is unbranched. There you go. So chitin keeps the tracheae open in the tracheal system of gas exchange in an insect. Gas exchange does not occur in the tracheae. Explain the importance of one adaptation of the gas exchange surface in the tracheal system of an insect. So a lot of people here were just talking about spiracles, and they didn't talk about what actually happens at the gas exchange surface. So what that means is where the actual exchanging of gases is happening, right? It's not talking about spiracles. Spiracles are just holes in the surface of the insect that let the air actually get in and out. They're telling you in this question, it's an adaptation of the gas exchange surface, where CO2 is going out, O2 is going in. So whenever you're having a gas exchange surface, what you want to do is you want to have the maximum amount of diffusion possible, because we rely on diffusion to do gas exchange. In our lungs, it's the alveoli, CO2 out, O2 into the blood, right? So what we need to do is think what maxes out the rate of diffusion. So rather than memorizing separate answers for every single animal and organism in the universe and type of thing, why not just think of the general principles and understand? So if we're trying to diffusion max, if you will, we need to first of all have a high surface area to volume ratio, right? And if we think about it for a second, well, they are branched, aren't they? The tracheoles, not the tracheae, the tracheoles, because that's where the exchange is actually happening, are highly branched. That's one thing. Next, what I need to think in my head, what are the features max out diffusion? Short diffusion distance is one of them. So short diffusion distance. So then you need to think, well, okay, well, we're in an insect. Where's the gas exchange system happening? It's the tracheoles. And we need to think, how does that possibly have a short diffusion distance? Well, the tracheoles are 1 cell thick. We can keep going because these tracheoles also enter directly into tissues as well. So there's even shorter diffusion pathway. Now, these are answers that wouldn't really be relevant here, but other things you can talk about as well, if we think about it, is concentration gradient. More specifically, we want it to be steep. So this, I don't think this is on the mark scheme, but a separate point you can generally make with, like, for example, humans and fish is that, first of all, the fish has a countercurrent system, but here as well, respiration, uses O2 and releases CO2, right? And also you've got the movement of the air in and out because of the rhythmic abdominal movements. That maintains that concentration gradient. And the last factor that you should be thinking about is temperature, which is irrelevant here. There's nothing you can do to max out the temperature. And if it's facilitated diffusion, then it's going to be the number of transport proteins. So rather than thinking, okay, I need to think about an insect and then just spew out facts. Understand the general principles. If we're doing gas exchange, all of our gas exchange depends on diffusion. What factors make diffusion better? Where is diffusion happening? The gas exchange surface, where is it happening? You have to do that in all your questions. Lignin is a polymer found in the walls of xylem vessels in plants. Lignin keeps the xylem vessel open as a continuous tube. Explain the importance of the xylem being kept open as a continuous tube. So here, less than 5% of people actually got all 3 marks, and a lot of people were just mentioning a water column and then said cohesion caused by hydrogen bonds, but no one talked about tension. And a lot of people were talking about answers to do with translocation and source and sink, which is not very helpful. Just as a quick side note on how water moves in plants, because this is something I see a lot of people struggle with. Here's my roots in my very sick drawn, epic plant, right? There's the leaf. Transpiration, people just cannot get the definition of this. Everyone I know so far, and I've taught, has always struggled with telling me what transpiration is. Transpiration is simply just the loss of water via evaporation out of the leaf stomata. That's it. It's nothing else. Transpiration then causes other things, but it itself is literally just water vapor lost. It evaporates into a gas and then diffuses out the stomata instead. It's not osmosis because it's no longer in a liquid form. It's diffusion. It's gas. So what happens is, because we have now put out water like that, it's going to create a tension. When you have a string and it's all saggy like that, and if you pull it and it becomes taut and tight, that is tension. You're pulling it. So it creates a pull, right? The reason it creates a pull is because all the water is one singular column here. Because of cohesion. The name tells you what it is. Cohesion tension theory. Because of cohesion, together, adhesion, sticking, The water molecules stick together because of hydrogen bonds, and they form one big long column. Here, because water's being evaporated, it creates a tension and yanks it up. Imagine here, this is a string, right? And we have this long string going through here. If you pull on this string, then this whole thing's going to get dragged up. That's literally what's happening in the transpiration process, leading to cohesion and tension. So the cohesion tension theory is transpiration causes the loss of water through the stomata as water vapor. As a result, this creates a tension which pulls on the column of water. The column of water forms because of cohesion between water molecules because of hydrogen bonds. And what that does is it actually pulls the water up through the entire xylem system from the roots to wherever it needs to be in the plant. That's how it works. So a lot of people just get confused on that. That's me clearing it once and for all. So here it says, explain the importance of the xylem being kept as an open continuous tube. So if it's an open continuous tube, it means that it's just a tube with no interruptions. Whereas if it had like this and this, then that can cause problems. So you need to think in your head, why is it useful to have just a continuous open tube? Well, if we have this water column trying to get through it and we have like breaks in it, then the water column is going to break. So if I have like water here, for example, and a bit of water there, and then water here, if that gets yanked up, that's not going with it. That's not going with it either. There's no cohesion or tension between, there's no cohesion between them, no tension for them. So what it does is it allows an unbroken water column. And you need to explain why that's helpful and why this happens. What happens is cohesion due to hydrogen bonds causes water molecules to stick and form a column. Then transpiration causes loss of H2O from the stomata as H2O evaporates. And what this does is it creates a tension which pulls the water column through the xylem. If we had a non-continuous tube that had breaks in it, this wouldn't be possible, like I've just described there. So that's how this question works. It's really important to understand what each term is, and rather than just sort of memorizing rote-learned answers. There's nothing to do with source and sink here, because that's to do with translocation, which is different. The human disease malaria is caused by an infection with single-celled eukaryotic organisms. Figure 2 shows a diagram of Plasmodium vivax, one of the species that can cause malaria. So if we have a look, we've got structures that contain enzymes that allow P. vivax to enter human cells. We've got a whole load of things going on here. Fine. It says here, other than the Golgi apparatus, name one structure in Figure 2 that shows that P. vivax is a eukaryote. So here, there's a lot to pick from. You just got to pick like a membrane-bound organelle. So that's an organelle that has its own membrane, right? So we've got mitochondria chilling here, and we've got like vesicles coming out of here. We've also got our rough endoplasmic reticulum here, and we've got our nucleus there. So any one of them works. So I'm just going to go with mitochondria. Mitochondria. Then it says, describe two functions of the Golgi apparatus in a eukaryotic cell. So here, people were just saying processing lipids and producing glycolipids. That's the same answer. What it does is it will modify proteins into glycoproteins. And what it does is it makes vesicles that allows them to actually get out and move around. A lot of people were saying, oh, ribosomes are also made here too. Ribosomes are not made there. The ribosomes are made in the nucleolus. RNA, so ribosomal RNA is transcribed to make ribosomes. That's where it happens, the nucleolus. So here, what we do is I'll say it's going to modify proteins, right, to form, like, for example, glycoproteins. So glyco, sugar, proteins, sugar proteins. So they're putting glucose on them. Or what it does is it forms lysosomes, for example, which contain lysosomes that break things down, or it could form vesicles. And vesicles are useful, for example, if you think about like nervous system, when you've got transmission of acetylcholine, what happens is vesicles are packaged and produced, and then they move across the cell surface membrane and move out the cell via exocytosis. So this is pretty important. So P. vivax evolved from a common ancestor in Africa. As humans migrated around the world, new strains of P. vivax evolved. P. vivax is now extremely rare in Africa, but there are several different strains of P. vivax in other parts of the world. Figure 3 shows a phylogenetic diagram of the evolution of these different strains. Fine. If we have a look then, we have this African common ancestor. From here, we had some division into this and this. Then from here, we had something else that divided into this and this, but also divided into Europe strain E1, and then so forth. Fine. Now it says, what does Figure 3 show about the order of human migration out of Africa? So here, you've got to use what the question's told you. It's told you that there's a common ancestor in Africa, and as humans migrated around the world, new strains emerged. So if the new strains have emerged, that's indicating when they were migrating. So every single time you got a branching off here, we've got new strains and stuff. So here, this is time. So we're looking for, when the earliest one is. So here we have the African common ancestor, which then becomes a different common ancestor in here, and a different one in here, right? So if we have a more detailed look now, here we have our common ancestor coming along, and then here later on, because remember this axis, it's not really an axis, but like this part is time. This is sooner than that, right? So we've got another common ancestor coming a bit later. So this one came first. And then from there, we had India first coming along. So that means that the first place they must have gone to, because the questions told you that new strains developed as they sort of migrated. First one is India. Cool. Then let's have a look. So here we had our second, like our first branch, if you will, from here. And here we had our first, which came earlier. So then if we move forward, the second one came here, second one came here. We can see that there's this new strain here, Europe first. That's still not from the second common ancestor sense and came sooner. So because that came sooner, the next place they must have gone to, Europe. Next, so we're on our second division, if you will, from there, second divergence here and here. We need to compare the two. So here we have East Asia coming first and then India again, but they've already done the India merge. So here we have Central America, but you see that's diverged a little later. So that means that East Asia came East Asia. Then we have this branching off here, which led to the South America strain, but we can see that Central America came out first. So that means that Central America first and then South, like that. Right. So the way to think of it is that these things are nodes. That's where common ancestors are. And these things are the tips, the leaves, sometimes people call it. That's where, this new thing that descended from it came from. So each branch is like that. So for example, here, that's that's like a grandparent and that's like your parents, for example. But there you go. So there are an estimated 229 million cases of human malaria each year. 94% of these are found in Africa, but not caused by VVAX. PVVAX does cause 61% of the cases of human malaria outside of Africa. Use this information to calculate the number of cases worldwide caused by PVVAX each year. 6% not in Africa, not VVAX. So if we work out 6% of 229 times 6, we end up getting 1374000, right? That's the number of cases that are outside of Africa. It's told you that it does cause 61% of these. If we do 1374000 times 61%, which would be 61 over 100, we end up getting 8381400, right? If we put that in standard form, it ends up being 8.38 times 10 to the... And there you go. In Africa today, most of the human population are resistant to malaria caused by VVAX. Use your knowledge of natural selection to explain why this resistance is so common. So here, a lot of people were just saying random stock phrases or rote learned answers or model answers. And they were like, variation exists. People with favorable alleles survive and reproduce and pass on these alleles and allele frequency increases. You cannot do that. You got to stop doing that. A lot of people were saying resistance gene, resistance allele. People were mixing the word allele and gene together. Some people were saying desired characteristics and traits. And then some people were even saying that mutations happen in VVAX as well. People were talking about immunity and people were talking about active and passive immune responses. Like you guys were just yapping over absolutely nothing that's relevant to this question. Here, the questions told you, use your knowledge of natural selection to explain why this resistance is so common in Africa. You need to look at who's being resistant. The human population are resistant to malaria. Something has changed in the humans to make them resistant to malaria. So natural selection is where you have a selection pressure due to nature. Something in nature wants to kill you and stop you from surviving and reproducing. That's what selection is. And natural means it's something in your environment that's doing it. If you can't survive and reproduce, you can't pass on your alleles that make you you. So you're gone, you're finished, done, right? But some people, just by pure chance, these mutations are random. You can't predict them. By pure chance, some people just happen to be, better at overcoming that pressure, right? So that person who has that allele is more likely to survive and reproduce and pass on that allele. Because all the other geezers that don't have this allele are also going to die and they ain't going to have kids. Your lineage is the one that survives, the one that has that allele. So over time, more and more people have this allele. And as a result, you see more of it in the population. Its frequency increases. It's not gene. You have the same genes, the allele is just different. For example, if you have blonde hair and I've got black hair, then we both have a hair color gene. We both have hair color being produced. It's the allele that's different because my hair color gene is still there. The allele is just black, whereas your hair color gene is still there, but it is blonde. So that's how this works. Here, we're going to talk about the steps that lead to this. So first of all, a random mutation leads to an allele causing resistance to PVVAC. You need to tailor this answer to the scenario at hand. The question has told you that most of the people are resistant to malaria, and it says use your knowledge to explain why this resistance is so common. The way they phrased this must have been to catch people out of their model answers. Model answers don't work. Just don't do it, right? So that happens. Then people with this mutation are more likely to survive and reproduce versus those who don't because they're not going to get infected. Now, as a result, you can't just go ahead and say desired characteristic on this garbage. No, What happens is the resistance allele, you've got to tailor your answer here, remember, is passed on to offspring. And then what happens over time, the frequency of the resistance allele increases. You have to tailor your answer. So for example, if the squares are people who don't have it, and then like that one circle is the geezer that does, right? If they all get infected by malaria, dead, 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 dead. Ooh, I'm alive. So let's make another one, right? These two will reproduce and survive. And then can you see how all of their offspring, maybe a couple might not, but like, all of their offspring have it. Skeezer dies, and then they all reproduce, and so forth, until eventually most of the population has that allele. That's how this works, right? Otherwise, if they didn't have some sort of selective advantage, then we'd have a load of squares here too as well, but they don't. So that's how this process works. Some hospital patients suffer from diarrhea caused by an infection with the bacterium Clostridium difficile. The C. difficile bacteria releases toxins. These toxins cause diarrhea. The toxins damage the cells lining the ileum, causing them to lose their microvilli, and the damage to the cells reduce the absorption of the products of digestion and reduces the absorption of water, resulting in diarrhea. Explain why the damage to the cells in the lining of the ileum reduces absorption of the products of digestion and why this reduces the absorption of water. There's a lot of hints in this question that are telling you what's going on, and people just kind of didn't listen to it. And a lot of people are just making very vague answers like water moves into the ileum and out. It's all in the question. It says, why is the damage to the cells lining the ileum reducing the absorption of the products of digestion, and why this reduces the absorption of water? So it's a two-step question. Here, they've told you in the question, it damages the cells of the ileum, causing them to lose their microvilli. This damage then reduces the absorption of the products of digestion and the water. So they're telling you to fill the gaps in. Damage to cells leads to lose microvilli, leads to less absorption of digestion and less absorption of products of water, causing diarrhea. So if we think about what happens in the normal process, here's your lumen chilling, right? There's your ilean epithelial cells that have their nice little villi and stuff, and then that's your blood, right? Now, if they have damage here, you need to think in your head, what's going to happen? Well, microvilli increase the rate of diffusion/active transport or facilitated diffusion because of the fact that they have that bigger surface area to volume ratio. So if that's gone, then you're going to absorb the products of digestion a lot slower. So it's all chilling here rather than getting in here, right? The questions also told you that you're absorbing less water as a result for some reason. So you need to think, why is there less water going inside here? Well, the water's chilling here. It can't get inside for some mysterious reason. What's happened is that because these soluble things like amino acids and glucose and stuff aren't getting into these cells, the water potential is not dropping in these cells. And in fact, the water potential is actually dropping in the lumen. So what happens is water accumulates here, and maybe even some might move out from the cells into the lumen, which is where, remember, the food pipe is, well, it leads to eventually. And now you've just got loads and loads of water in here. Eventually, that's going to go through your large bowel, and you're going to shit it out. And that's where you get diarrhea from. So that's what happens. It's a similar process with lactose intolerance. Because you can't absorb or digest the lactose, it actually accumulates in here, and as a result, lowers the water potential in your bowels. So then water moves out into the bowels and causes diarrhea. Same principle here. So we need to talk about each and every single step leading to this. So then what happens is you've got fewer microvilli, which leads to less surface area for absorption. What happens then is your digested products remain in the ileum, and that lowers the water potential. Obviously, you write in full. I'm just doing it quick like this. So then what happens is water moves out of cells via osmosis into the ileum. Or you can say the flip side. You can say less water moves into the cells, out of the ileum via osmosis. You have to say the journey. You need to say by what means things are happening. It's all hinted at in the question here. You can't say water moves in or water moves out. You need to say by osmosis. You have to say what means any movement's happening. Generally speaking, if you're talking about particles moving, you have to say diffusion, active transport, facilitated diffusion, osmosis, whichever one it is, or simple diffusion. Not all patients in hospital with C. difficile develop diarrhea. Scientists measured the antitoxin antibody concentration in hospital patients with and without this infection. What they did is they measured the antitoxin antibody concentration 4 times. So that remember that the C. difficile makes a toxin that causes diarrhea. This is an antibody that's against it made by people. So what did they do? They measured it 4 times on the day of admission to hospital, so day 0, 3, 6, and then the day they left the hospital, and here's their results. So whenever you're doing these kinds of questions, I always think it's helpful to look at independent and dependent variables. I just force it on there to make sense. So if we look at independent, what's changing is they're doing, well, if you have a look, it's C. diff, no diarrhea, C. diff, and diarrhea. So whether it's C. diff or none, and then diarrhea or not. And the dependent, right, is what they're measuring. So it's very tempting to just say, the effect of the immune response. Look at what they're measuring specifically in the question rather than just what the stem of the question is telling you. So here, they're measuring the antitoxin antibody, and this is the mean amount in some mysterious units that they've come up, arbitrary unit. Is there any information we can get from here as well? That's about it. So then if we have a look, just describe to yourself quickly what's happening. So here, this is C. diff but no diarrhea. So the dark squares. If you have C. diff and no diarrhea, we can see that the antibody concentration's up, and then it starts to level, right? It hits the peak at about day 6, and then at the day they leave the hospital, it's a bit down. Fine. These lot had no diarrhea, but they did have C. diff. Fine. Now, the next one is people without the C. diff infection. So without the C. diff infection, their mean concentration was just 1. It kind of went a little bit down and back up. So there's no infection. And then here, these lot had C. difficile with diarrhea. So C. diff positive and diarrhea. So we can see here that their mean antibody concentration kind of went down at days 3 and then kind of slowly climbed up and then kind of leveled off. So if we compare them, between each of these, one variable has changed. So here, they have C. diff and they have diarrhea. These are C. diff and no diarrhea. So the only difference between these have way more antibodies than these, and these had no diarrhea, these did. So there must be some link, if they've done this experiment right, with antibody concentrations and diarrhea. From this data alone, you can say that if you have more antibodies, you're not going to get diarrhea, whereas if you have less, you're going to get diarrhea. And if you have no infection, your antibodies don't do anything, which is what this experiment results are suggesting. So you need to get in the habit of just being able to do that rapidly so you can make sense of what's going on. Otherwise, just don't. What are you doing, you know? So the scientists suggested that the antitoxin antibody could be given as a form of passive immunity. Use figure 4 to suggest how this passive immunity would work and which patients should be offered this. So a lot of people didn't understand how to apply this to the data here, and they just gave loads of lengthy model answers, again, talking about antibodies neutralizing bacteria or destroying the pathogen. You can say these answers, but you got to tailor it to what's going on here. A lot of people just had no clue what passive immunity actually straight up meant, and they were talking about clonal expansion. Active means you're doing something. Passive is where you're not doing something. If you're passive and you're just watching things slide by and chilling, that's what it is. Here, active immunity means you've had the antigen. Antigen means attack generator, so something in your body that makes an immune response. The only thing that stimulates an immune response is an antigen. So you've had an antigen in your body that made you have the immune response, which means you did the phagocytosis, the helper T cell stimulation, the B cell stimulation, the clonal selection, blah, 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 all of that stuff. That's you did it actively. Whereas passive, you didn't do that. And instead, you're being given these antibodies. The differences are that active means you're going to make memory cells and you can persist that, sort of immunity for the rest of your life or maybe for a long time. Passive, the moment those antibodies are finished, they break down and they're gone. You don't have them forever. Questions told you it's passive. They've told you to say how it would work. And they've also told you to say who would benefit most from this. Here, first of all, we are talking about antitoxin antibody. Toxin. It's against the toxin, not the pathogen. So how it works, first of all, is that it's an antibody that destroys or causes the destruction of the toxin, not the pathogen. As a result, you need to look at this figure and say what the conclusion would be. If you have more of this antibody destroying the toxin, we can see here that you get no diarrhea. That's what the data shows you. That's why it's telling you use the figure. So here, that means you'd get no diarrhea, right? And then it says which patients would be best offered this. Well, you can see here, you don't want to give it to people without the infection. These lot have C. difficile and no diarrhea. There's not really any point in giving them, and their antibodies are quite high. Whereas here, these lot have an infection and diarrhea and barely any antibodies. Based on this figure alone, if we dashed them some antibodies, maybe they won't have diarrhea. So here, then, you would offer to C. difficile patients with diarrhea. And then why? It's because they have low antibody concentrations. There you go. To be used as a passive immunity treatment, the antitoxin antibody would be injected. If it was given by mouth, it would be digested. Describe how the antitoxin antibody would be digested. So here, a lot of people some of people just weren't even talking about digestion at all. Some people didn't remember that antibodies are proteins, and others just described absorption. Stop using model answers, bro. And also a lot of people were just saying GCSE level stuff. They were saying bonds broken. No one was talking about just what's going on, The question has told you, literally says, describe how this antitoxin antibody would be digested. From the mouth, what happens? This is a protein. If you have a protein, when you're digesting something, you are breaking bonds, but you cannot just say breaking bonds. You need to say what you start with, what bonds are being broken, and what you're making. So within this, it's a protein. So what happens is that peptide bonds are hydrolyzed. Not broken, hydrolyzed. But you can't use hydrolyze all the time. For example, you can't hydrolyze hydrogen bonds in DNA helicase. That's another mistake I see people say a lot. Here, these are atoms and molecules bonded together, with a water-gone condensation. So this is hydrolyzed. So here, peptide bonds are hydrolyzed, but that's not all, because we're talking about the full digestion process. When we're doing that, we have a long protein here, right? It's a quaternary protein, it's huge. We have specific enzymes that will break this part and break this part at the ends, and then ones in between. So those are called endopeptidases and exopeptidases. So endopeptidases break, peptide bonds. You could say break now that we've said high peptide bonds within the antibody. And then exopeptidases, I'm just going to episode for short, break bonds. We could say terminal bonds if you want as well at the end. So peptide bond of the antibody, right? AB for short. And then we're left with dipeptides because we can't, once you have this and this, which one breaks it down? Is it going to be an endo or an exo? You can't tell because there's no in between, there's no ends. So what happens is then the last step of breaking down a protein is you have membrane bound dipeptidases. And what they'll do is they'll break down the dipeptide into anyone? Amino acid. You have to say the full journey like that. That's all they want. They don't care about absorption. The question is literally saying digestion. So to summarize, you have huge protein. What happens is you have endopeptidases and exopeptidases hydrolyzing peptide bonds within, the molecule and at the ends. And then what happens? You're left with a dipeptide. This dipeptide is then broken down by membrane-bound dipeptidases. These are enzymes that are membrane-bound. They're stuck to the membrane of the ilean cells, and then it's broken down into amino acids. You have to say all of that. This should be free mark. If you didn't get, all marks on this, you've got to get that bread. You got to get it up. You got to lock in. A student investigated the use of cinnamon oil as an antimicrobial substance. She investigated the effect of cinnamon oil on the growth of 5 different antibacterial cultures grown on agar plates. They added 100 millimeters cubed of each bacterial culture from its glass bottle onto a separate agar plate. They then spread each bacterial culture evenly over the agar using a spreader. Describe how aseptic techniques should be used. Well, sorry, which one she should use. So here, a lot of problems came from just illogical and lengthy answers that had no relevance here, because all they're saying is to just literally say, what are they going to do? So a lot of people are saying flame the lid, flame the agar plate, sterilize surfaces, and upward air movement kills bacteria. And a lot of people were saying things like hold the plate away from the face, wear gloves, work quickly, close the windows. I don't know why you'd want to close the windows of all things, but unless you're trying to get stuff coming in, I guess, for contamination here, it's like aseptic techniques that she should use for her example here. You can't just say general steps that you've memorized. It literally just never works. Another problem, though, is that a lot of people have said really illogical steps, saying using a loop to spread 100 millimeters cubed of culture. 100 millimeters cubed is about a third of a Coke can. That's a lot of volume for a spreader to use. You're not putting that much bacteria on a plate. So just think for a second what you're doing and saying, and don't just jump into yapping random things. Number one, here, they've added this much bacterial culture from a glass bottle onto a plate. What you need to do is you got to take it out of the bottle and you've got to put it on the plate somehow, right? And then once you put it on the plate, you don't want it all just concentrated here. You want to spread it out. Think about it step by step. So number one is to get it out there and put it on there. You use a sterile pet to transfer bacteria onto the plate. All right, back for short to plate. Then number two is you want to spread it out. The spreader doesn't put it on there. It's actually spread it. So what you do is you use a sterile spreader to spread the bacteria. Now here, they've told you that we're using different agar plates and there are 5 different bacterial cultures. You don't want to just stick the same pipette back in the next bottle or you just contaminate the next one. So then what you would do is you would immediately disinfect the pipette and spreader, unless you're using a different one, then you would say use a different one after use by, I don't know, dipping in disinfectant. If you just think about it logically in them steps, you've already secured the bag with 3 marks. But what else can we say? Well, all the other things you could say is you could flame the neck of the bottle as well, because if you sort of accidentally contact the bottle, something you might contaminate like that. As well as that, you should lift off the agar plate at an angle so you don't, again, fumble around and get all the bacteria in there as well. And as well as that, if you work close, then you've got upward air movement as well, which prevents contamination rather than killing bacteria. So on each agar plate, the student cut a hole in the agar. The well had a diameter of 6 millimeters. The student added 50 millimeters cubed of cinnamon oil into the well. Calculate the minimum depth of the well to allow the addition of 50 millimeters cubed more of this oil. Use the following equation in your calculation and use 3.14 as the value. Hi. So here, this is another unfamiliar question style. No one's seen this one before, but it's doable if you just sort of break it into chunks. All these maths questions, my advice is if you can't do it, leave it. It's 2 marks, not worth it. But if you going for it, just break it into steps. Here, we need to work out the minimum depth of the well to allow this. You've got to make sense of what the depth means here, right? Now, told you that they've cut a hole in the agar and it's a cylinder, and they've told you some features about it. So it's cylindrical, and the stuff is going in this hole, right? They've told you that it's got a diameter of 6 millimeters. Cool. The student added 50 millimeters cubed of cinnamon oil. This is a volume. They want to put enough cinnamon oil in this so that it's nicely filled. Right? The question's asking you to work out the depth of the well. They're asking you to work out this bit here that would allow you to fit in this much cinnamon oil, which is 50 cents cubed. So that means in order to fit that in, the volume of this well must be 50 millimeters cubed, which they've told you is a cylinder. So that means that 50 equals pi r squared L, right? Now this is L, the depth, which is what we want. So if I just move it here, 50 divided by pi r squared is going to equal L. Now, r is the radius. They've told you the diameter is 6, so r is 3. So that means that L equals 50 divided by pi times 3 squared. But they've told you to use 3.14, so it's going to be 50 over 3.14 squared. If we do that, we get 1.769. Let's do it to the same number of significant figures they gave us, 1.77. There you go. So the student kept the plates at 25 degrees Celsius for 24 hours, and then figure 5 shows what one of the plates looked like. So here we've got bacterial culture growing on agar. Here's the well containing the cinnamon oil there, and we have a clear zone here with no bacterial growth. So on that figure, based on that figure alone, this cinnamon oil might stop bacteria from growing here / kill them. She then measured the diameter of the clear zone with no bacterial growth around each well, and then made these measurements to the nearest whole millimeter. So the results are shown here. Here, what we should do is scribble out what our independent and our dependents are, right? So there's a couple. The independent here is, first of all, the species, but also the cinnamon oil, right? And the dependent is the diameter of the clear zone. Now, the bit that's confusing is you have the positive and the negative control. Here, we have some mysterious thing that's a positive control and it produces a clear zone. So a positive control is a control where you have this experiment running where you know the outcome is going to happen. So for example, here, what they've done most likely is they put an antibiotic they know will cause this specific, clear zone that will cause that will kill bacteria. So that, for example, here with this bacillus species, they've used an antibiotic that they know will make this be 14 appear. So here they've used an antibiotic of some sort that is known to do that. That's what the positive control is. You're putting a control thing here where you know the outcome already, almost, right? So for example, if I did an experiment where I don't know, I had 10 people and I gave them chocolate, the classic shit yourself one for those that watch my other videos. This would be giving them the chocolate, which makes them shit themselves. The negative would be they don't get chocolate, so they don't shit themselves. And my positive control would be, I don't know, me lacing the chocolate with diarrhea-inducing toxins that make them shit themselves. So I know they're going to shit themselves if they have it, if that makes sense. So it's the same principle here. You're putting an outcome that you definitely know is going to happen. Now, if we have a look here, what's going on? So it seems like Staph aureus seems to be most susceptible to cinnamon oil. Positive controls, whatever. Negative controls, all 0, as you would expect. They're all happily grown. Chill. Fine. Oh, and would you look at that? It says suggest exactly what the student added to the wells to get positive and negative control results. So a lot of people were saying doing nothing to the well, that's incorrect. And then people were just saying adding bacteria to the well as well. Are we not doing that with every single one of these? Come on. Look carefully and think carefully about what's happening. Let's do our negative control. That one's easier. The negative control is the thing that's not, your experiment's not there. Like the thing that you're adding that's funky and cool. So that here, what you're doing is you're adding bacteria, but no cinnamon oil. Both of these are controls, so they're not going to have the cinnamon oil because that's what they're investigating. So for our positive control, we're adding bacteria, no cinnamon oil, because, it's a control, but we're adding something we know will definitely cause a clear zone. So it must be an antibiotic or an antiseptic, because that's going to cause that outcome. That's what positive control is. Then it says complete table 1 to show the mean and median diameters. So remember that the median is just the middle value. So what you do is you go up in order, from smallest to largest, and you just plonk the middle number in. So here I'm just going to skip ahead and do that. So it's going to be 16 and 12 here. And then the mean is you add them all up and then divide by the number of them, just 17 and 30. So the mean plus or minus 2 standard deviations includes over 95% of the data. Use this information to consider whether the standard deviation suggests the differences in means are likely to be due to chance. So what we've got to do is we've got to look at all of the bits and bobs here. So if we have a look, they've given us the standard deviation of all of these cultures, and they've told you that 95% of the data is within that range. And if we're looking at likely due to chance, what that means is, did they get lucky with the result, or is there actually a difference? So for example, if I flip a coin 10 times, you would expect it to be 5 heads, right? Generally, but not everyone gets 5 heads. Sometimes you might get 10 heads, and that's by pure luck, by pure chance. When we're doing statistical testing like this, we're looking to see if it was just luck, pure chance that something happened, or if there is possibly an actual difference. So all of these statistical tests, like the p-values and all of that stuff, what they do is they work out the probability that the results are due to chance, as in the probability or how likely you are to have just been lucky. So for example, if you worked out the p-value for my 10 heads magic coin flip, I probably just got extremely lucky. So the p-value is probably like really higher than 5% that we're used to. That's just telling you the probability of the results, whatever they are, being due to chance. So here, let's have a look what's going on. So it says the mean plus or minus 2 standard deviations includes 95% of the data. When we're talking about SD overlaps and this and that, blah, 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 it has to be this, the mean plus or minus 2 standard deviations, right? In the table, they've given us just the standard deviation. So we actually need to do the mean plus or minus 2 times this. So mean plus or minus 2 times 2.4, for example. Right? So if we just add an extra bit here so here what we need to do is we've got to work out the mean plus or minus. So let's do the plus 2 times the SD and then the mean minus the 2 times the SD, right? Because that's what we're trying to look for, is there overlaps between these. If we do that, we'll have this plus 2 lots of that and then this plus minus 2 lots of that and so forth. So here for this first one, we would have 21.8 and 12.2. Here we would have 17.4, 8.6. And then obviously here, it's 0 is 0, right? So then it says, use this information to suggest whether they are likely due to chance. So, the SD overlap thing that we love so much, now we can have a look to see if there's any overlapping between them. You can see that the mean plus or minus the SD's overlap. This is from 21.8 to 12.2, but here it's 17.4 to 8.6. Because they overlap, that means there is probably results. These results are likely due to chance, but you can't just say results. Literally, like every mark scheme says, you cannot say results are due to chance. You have to say what it is here, right? Here, they're asking, is the difference due to chance, the difference in the means? Because they overlap, they are. So you'd write the calculations, obviously, you'd plonk here, and you would say that these SD overlap. Therefore, the difference in mean clear zones are likely due to chance. Because if we think about it like this, right, here's one set of results, here's another set of results. Can you see how there's overlap between the two of them? They're not two separate entities, right? If I had here and here, and then here and here, there's not much, they're very two different chunks. But the absolute lowest relative to the mean that's, covered in this range doesn't overlap with here. So they're probably two separate entities. That's what this whole overlap stuff is. The reason we do plus or minus 2 standard deviations is because it gives you 95% of the data. That's a pretty decent amount of data to include. So if 95% of the results from here and here overlap with each other, then it's probably going to be due to chance because there's similar results between the two of them. That's what it's trying to get at. So it says define the genome and the proteome. Here, a lot of people were just saying genetic information, the genetic code, the genetic constitution. That's absolute, come on. No, it doesn't, that's not what the definition is, and that's just very vague, and there's no precision in that. Here, what it is it is the complete set, or all the genes, it's a complete set of genes in a cell. Every single gene, right? For example, in like your mouth, you've got your genes that make like salivary amylase, saliva proteins, all of that stuff, right? Those are every single gene possible that is there in that cell, right? The proteome, this is another one that people just kind of said. They were just saying proteins made. Proteome is expression of proteins in the cell, or all the proteins a cell codes for. You've got to say specifically what it is. It is the full range of proteins a cell can produce. The proteome of your stomach, for example, is going to be mad different to stomach cells, is going to be mad different to the one in like your hands, like your skin, because some of those make hydrochloric acid. If you made hydrochloric acid in the proteome of your skin on your hands, you'd burn your skin and you'd die. So it's really important we have that very specific thing. And here as well, some people were saying range of proteins that a species or population can produce. No, what are you doing? No. The classification system used in the early 20th century grouped different species of bacteria according to the position and shape of the flagella on bacterial cells and by the number of flagella per cell. They were observed using an optical microscope. Each species of bacterium had a characteristic cell shape and arrangement of flagella. These characteristics may be shared with other species within a genus. Flagella are fragile, difficult to stain, and may extend from the cell at any angle. Consider the accuracy and limitations of the early classification of bacteria using the arrangement of flagella. Here, we're talking about how accurate this is and what the limitations of it are, right? So we need to look through this text that they've given us, the context, and use the context, not just say what we've memorized. A lot of people were just saying memorized answers, and it just doesn't work, right? First of all, they're saying that they classified bacteria by looking at them and seeing how many flagella each cell had. Immediately, optical microscopes are not very good. Optical microscopes have a low resolution. You can't just drop that. You've got to say what that consequence has in this context. It might be difficult to tell apart different flagella. You don't know if there's two mishmashed ones or if they're different. So for example, here, if I draw these two dots very close together, you can tell they're apart. But if I zoom out really, really, really far, can you see how they look like one dot? Low resolution. The resolution is the ability to tell apart two very close together, and you can't do that in that example, right? Now, what else is there that could cause problems, if you will? Here, we can see that the questions told you that they are fragile, difficult to stain, and may extend from any angle. Like it's literally giving you the goods there. You have to talk about the consequences, though, as a result. If they're fragile, then remember, think about how you prepared like a microscope slide. You've got to do all of this stuff. You've got to stain it. You've got to squish it in the thing and push it down and this and that, blah, blah, blah. Like, if fragile, you're going to get broken flagella, aren't you? And then what you need to talk about the consequence. Well, you may not see it. If you can't see them, they're not going to count. Other things you can say is that they're difficult to stain, so they're invisible. Other things as well is that if you're doing it at an angle, you might not be able to see them. And as well as that, classification of bacteria, like not all bacteria have flagella. So there you go. So it says suggest why several bacterial species have been renamed in recent years. Here they're asking you, why have they decided to change the name? So bacterial species, you need to think, how do we name species? What's it all to do with? It's all to do with phylogenies, domain, kingdom, blah, 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 genus, species, right? So something along the way has changed over time in recent years and made them think, oh my God, we need to change this up. In the previous question, they even hint at it a little bit, telling you, look, they used to do this and it wasn't very good, right? They used to look at light microscopes, used to look at the number of flagellae, and that was what they did. But now we don't do that anymore. And for some reason, so it's different. A lot of people then were like, got to that conclusion. They were like, okay, not bad, not bad. And then just said new technology or scientists are better at classification or the previous method was inaccurate. Other people were saying that they'd evolved into new species and new mutations had happened. That's not what they're saying. Here, what we need to do is we need to look at what's going on now versus then. The way we classify organisms is, for example, if we looked at the microscope one, we now have electron microscopes, so we can do it better. So if we did use that, we can do it that way. But what we now tend to do more so is we have amino acid sequencing and DNA base sequencing. You can't say genome sequencing. It's the base that needs to be mentioned because what we're doing is we're comparing the base sequences. If you have two organisms with lots and lots of similar bases, they're probably quite closely related. Whereas if they're really, really different, they're probably different. So here, simply just by saying, I don't know, DNA base sequencing is now used or something like that, it's probably enough because now as a result, we're able to tell much better rather than just looking at them and hoping for the best. So here it says figure 6 shows an image from an optical microscope of a single bacterial cell. The bacterial cell is 2.3 micrometers long, excluding the flagellum. Calculate the magnification of this image. Show you working. So I can't do this because the rule is a bit janky. But use the formula I equals AM. I am. Image equals actual times magnification. Magnification equals image over actual, right? You need to make sure that the units are consistent though, right? So what you would do is you would measure they've told you it's this long. So from here to here with your ruler, which would be in centimeters, convert it to micrometers, or you convert this to centimeters, either's fine, and then you just do it and you're done. Bish, bash, bosh, finished. Carbon monoxide is released during incomplete combustion of fossil fuels. Figure 7 shows the dissociation curve for oxyhemoglobin when not exposed to CO and then exposed to CO such that the 50% of the oxygen binding sites are occupied by CO, 50% CO hemoglobin. Fine. Have a quick look at what's going on before jumping into random bits and bobs. So here, I almost like to think of an independent site type thing. No carbon monoxide, 50% carbon monoxide. So hemoglobin that's got, 50% of it's been, snagged by carbon monoxide, free to do whatever you want. This looks pretty happy. It looks like your standard hemoglobin dissociation curve. Whereas here, it kind of goes up and then it just stops. It's no longer getting saturated, even though the PO2 is getting higher. More oxygen, it's leveled off. It's not getting more saturated. This tells you how much hemoglobin has O2. You can see it's not going up. So this CO is clearly stopping it from being able to do that. Using figure 7, what can you conclude about how exposure to CO affects the loading and unloading of oxygen by hemoglobin and explain your answer? About 40% of people got no marks because here they were just completely recalling random bits of knowledge and hoping for the best. Cannot do that. Model answers do not work. I swear by this. I'll take this to my grave. It don't work. A lot of people were saying COHB would load more readily at low PO2, but that's not what they're trying to get at here. I mean, yes, fine, it does. While that's correct, great, but that's not what they're saying. They're talking about the exposure of CO2 affecting the loading and unloading of it as a consequence overall. So yeah, I guess there's that, but like, hello, there's this big flat line. Like, what are you doing? You know? And as well as that, some people were saying that it would not load oxygen at all. And a lot of people were saying the Bohr effect, changing blood pH, none of that's happening. Look at what's happening and then explain what's happening. Here, you can see that there's less oxygen being loaded than here throughout the entire process immediately. And also as well as that, the maximum you get here is 50, whereas up here you tend towards 100. So immediately describe what you see. The maximum saturation for the CO hemoglobin is 50% versus nearly 100 for no CO, right? Now, as well as that, we need to look at the things to do with affinity as well, because it's talking about loading and unloading and what's happening, right? The way I like to think of it is rather than left, right, and all of this, here's what I think. Here we're in the lungs, high saturation, high oxygen. As we go down this curve, we're getting lower and lower in the oxygen. So it's unloading. The saturation's lowering. So as you go down, it's unloading. So it's holding on, it's unloaded, bang, right? If I do this, I'm holding it on for a little longer and then I drop, I must be unloading it less readily, as in I'm holding onto it tighter, so I have a higher affinity. Whereas if I'm like, let go of it now, that's a lower affinity. So here, if we're comparing the two curves, can you see how it's holding on, blah, 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 here, and then if you think about it, pretend if it's up here almost, we can do it here, holding on, letting go there. It's letting go, it's dissociating like at a lower PO2. So it's holding on to it more, meaning that at a lower PO2, this CO hemoglobin has a higher affinity. It's holding on to the O2 for more, right? Let's describe what that means. It means that it has more O2 at a lower PO2. And that is true because if you look here, for example, if you look at where the saturation is 25, can you see it's about here versus here? So there you go. If you've got to relate it to this example here, it's always helpful to just say what you see on the graph. Even if you don't get a mark, just say it and then explain why. Why is this thing happening based on my knowledge? That isn't just a memorized answer. That would explain this. So the World Health Organization suggests that to avoid long-term health effects, the COHP concentration should be kept below 2.5%. They recommend that people should not be exposed to air with greater than 10 milligrams per meter cubed CO for more than 8 hours, or air with greater than 30 milligrams per meter cubed CO for more than 1 hour. Scientists have used a mathematical model to calculate the exposure of carbon monoxide that would result in 2.5% carbon COHP in adults and children, and here are their results. Again, just have a think what's going on. Independent and dependent, that helps massively. So our independence is obviously 1 versus 8 hours and adults versus children. And what did they actually measure then? So here, they worked out the concentration of CO in air that causes, turn it into your own words, that causes 2.5% HB. As in, what limit can you hit? In children, at 1 hour, at rest, the amount you need in the air to get them to hit that 2.5% is 31.2 milligrams per meter cubed, whereas in an adult, it's higher. So the adult can tolerate more CO before it hits that 2.5 threshold that they told you would cause health problems. Whereas in children at 8 hours, right, it's 9.6 versus 10.6. Fine. The scientists suggest that the WHO recommendations for carbon monoxide concentrations that cause a 2.5% COHB should be reduced to evaluate the conclusion. Remember that the WHO said that if it's greater than 10 for more than 8 hours, right? So I'm going to just scribble down here, greater than 10. And then if it's for more than 1 hour, it's greater than 30. So in a kid, it's greater than 30, it's greater than 40. Bingo. But if you look here at the 8 hours, in the kid, it's technically less than 10, but this is greater than 10.6. So why not just be like, why not just put it as like 9 point something, right? So with evaluating questions, you need to talk about positives, negatives, any problems with their method validity. So what I mean by that is what data directly supports what the statement is. Number 2, what data directly contradicts the statement. 3, does the research question actually, what they measure actually answer the question that they've measured? And is their method good and useful? Does it actually relate to this thing? And any other problems that can come into this as well? Because it's asking you, can you look at this and say, yeah, you've got to go through the entire context of the question. For example, I use this example all the time. If I have 10 people in a room and I want to test to see if, chocolate makes people shit themselves because of lactose intolerance, if I give 10 people in a room and they shit themselves and I get 100% shit rate in this sample, that means I can't just go ahead and conclude 100% of people shit themselves if they eat chocolate, because that's not true. There are plenty of you that's probably eaten chocolate in your life and haven't shat yourself. So you got to look and critique this experiment. So if you look at the positives that support this, 100% of my sample size did indeed shit themselves, which supports the statement. There's not really any evidence to contradict it. No one didn't shit themselves. All of the problems in that experiment lie with their validity, the methods, and what they actually did, all of that stuff, right? So here then, you would say, well, you've only done 10 people, small sample size. We don't know what chocolate they were given. We don't know if they have pre-existing lactose intolerant health conditions. We don't even know how much lactose is in the chocolate. We don't know how you pick these people. Did you just decide to pick people who shit chocolate and get good results? You've got to really narrow it down and see what's up. We also have no control. So I don't know if it was the chocolate that made them shit themselves or if it could have been something else. Like, I don't know, maybe they all had diarrhea from like a dodgy kebab from down the road. You got to think of it like that. So let's say what data directly supports their statement first, right? So what supports it? They're saying that you should put the thresholds lower. They said 10 is the threshold for 8 hours. Well, you see the kid is 9.6. So for the child, it was 9.6 concentration that caused the 2.5%, blah, 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 which is supportive, right? But let's talk about contradicting statements. What goes against it? For the adult, both 1 hour and 8 hours were higher than 30. Can you see that's, 30 and 10 respectively. So you can see it's 10.6 and 40.2. That's directly going against the statement, which is, they should put the threshold lower. It's fine, right? That's like the main bits and bobs there. But now we need to look at the problems with this statement, right? So the problems with validity and this and that, blah, blah, blah. So again, the statement is saying that the scientists suggested that we should make the thresholds lower. We need to look at the whole question. They said that these thresholds came along to prevent long-term health effects in people as long as the air around them is below that. They used a mathematical model to calculate them in adults and children. So immediately, number one is that a mathematical model may not be accurate. Why? Because it's not real people. You're never going to get, results as good as actually using real people. As well as that, what else is there? If we look at the data here, these are at rest. People don't just sit and do nothing. The results are only for at rest, right? And the other thing I like to think of, I should have mentioned before, is I always like to think, what could screw up this result? That's a good way to think about it to help you come up with problems, if you will. Because if anything's screwing this up, then the experiment's not good. Any experiment is where the independent is the only thing changing the dependent. So you need to think of factors that can mess up how much CO is eventually in the blood that causes that. So what threshold they have, right? Immediately, well, it's only at rest. If they're exercising, it may need less or more. As well as that, we only know there's adult and child, but we don't know anything about size, age, sex, ethnicity, we don't know anything to do with that. That might play a role. Some people might be more susceptible, others aren't. We also don't know about their health anyway. So, for example, if they have any respiratory conditions or lung conditions, that can make it harder. As well as that, in real life, we might not actually be able to get the air below these thresholds, for example. Maybe it's not practically possible to do that. That's why they have it. Maybe that's just what industrialized environments are like. As well as that, we have no stats test as well, so we don't know if the differences are significant. You cannot just say no stats test. You've got to say why. No stats test, but we don't know if the differences are significant. We don't know. People were just saying, we don't know if the results are significant. You can't say that. You have to say what the consequences, why you wouldn't want them. General rule of thumb is think about what's screwing it up. Say, why would I want this? What's it going to help me do? If you're saying stats test, you can't just say, we don't know if the results are significant. You need to think, why am I saying this? Otherwise, what's the point? We have to think of it like that. Otherwise, for example, if they went and lowered that threshold, that could cause huge amount of problems. People could die. So that's why we're very, very nitpicking. You've got to get in the skills of doing it like that. So scientists investigated a drug called MITMAB as a treatment for cancer. MITMAB inhibits cytokinesis. Figure 8 shows the drawings of cancer cells seen with an optical microscope from a sample treated with MITMAB and a control. So remember, just kind of go with it. So the drawings in A can be identified as those in MITMAB. Explain why. So just scribble it down real quick. MITMAB. No MITMAB. And you see, again, an independent variable is the only thing that's changing our dependent, which is what they're measuring, which is what the cells look like, I think. Probably, yeah. The cells in drawing A can be identified as those treated with MITMAB. Explain why. So here, a lot of people were just saying, cytokinesis is inhibited. You can't just say that on its own. It's saying, explain why this drawing is that. So you're almost there, but you've got to say why. Otherwise, how do you know? You need to tell them how you know that. Here, we've got our cells, but we've got 2 nuclei, and here, these are all 1 nuclei. Clearly, this cell can't split into 2. The question's told you, it's told you as well, where is it? It's inhibited cytokinesis as well. So link back, you can see that there are 2 nuclei in cells because there's no cytokinesis. There you go. There's not much else to say. Also, you've got to remember as well, these massive gaps for answers are just there because the exam board can't be asked dealing with all the extra papers, which I don't blame them, to be honest, because when I did my A-levels, I used so many unhinged amounts of extra sides of paper. So that's why there's just huge spaces there. It's not that you have to fill it all. MITMAB acts as a non-competitive inhibitor of an enzyme called dynamin. Suggest how MITMAB can cause dynamin to become inactive. A lot of people say that the MITMAB bound to the active site, which is incorrect. That's a competitive inhibitor. Non-competitive binds away from the active site. As well as that, people just straight up didn't talk about the consequence. They described every single step, saying the active site changed shape, and then just that was it. Nothing else. What's going on? Why aren't you doing that? You've got to say from start to finish what's happening. Otherwise, what is the point? Here, MITMAB binds to site other than the active site, or your allosteric if you want to be fancy, fine, right? What happens then is this causes, and don't say 3D, please, come on, every exam board mark scheme literally tells you off every time, give it up, stop. What it does is it causes the tertiary structure, which is where your active site comes from, to change shape of dynamin, and therefore it changes its active site. What happens as a result? You need to say what the next bit is. It's no longer complementary. That's it. Because otherwise, you're not saying why it's become inactive. The last step is saying why it's inactive. Otherwise, you don't get the marks. So inactive dynamin has 2 functions. It stimulates cytokinesis and inhibits cell death. Fine. The scientists treated actively growing cultures of cancer cells with MITMAB. What did they do? They incubated 1 sample of 2,500 cells with without MITMAB as a control, and then 8 samples, each with 2,500 and different concentrations of MITMAB. So what are they doing? Independent and dependent? Let's have a look. So their independent is MITMAB. concentration and also none, like they have some with, different concentrations and some with nothing, they're dependent. Don't just go ahead and say, like that number of cultures of cancer cells or investigations of it. Look specifically at what it is. Here, what did they measure? The proportion of control growth, not number of cancer cells. I didn't say that anywhere. Proportion of control growth. The reason you do that is because then you know specifically what's going on. The map should change the proportion of the control growth, as in the cells, like the percentage almost, if you will. So if the proportion's 1, it means that the number of cells, cancer cells in the map versus control are equal, as in they're the same. After 72 hours, what they did is they measured the number of cells and then they did this calculation. We don't know what the number of cells are. Otherwise, that's why I'm not writing it there. A negative value for the proportion of growth means that fewer than 2,500 cells were counted after 72 hours. That's a useful point. If we have a look then, what's going on? So at these concentrations, the proportion is 1, meaning that, like, again, it would be, whatever, some number over the same number as the control, as in there's no difference. Here, the proportion of control growth is lower, meaning that it grew less compared to the control, even less. So it means that across this point, the map has stopped them growing as fast compared to the control, which has no map. So it means it must be doing something here. And then here, it levels off, it stops. There's no more further decreases. So the amount of stoppage is the same here and here. Fine. It says use all the information given to explain these figure results. Again, a lot of people just got no marks or 1 mark for this because a lot of people were just saying more inactive enzymes, more cell death, and that was about it. Like, they didn't say why, they didn't say the trends, they didn't say it related to the figure, and, just what was going on. You have to talk about everything to explain this figure. So they were right to say that there's obviously more, less cytokinesis and so forth, but they didn't link it to the figure, right? Here, barely anything's happening. And that's, if you look here, that's an increase in concentration. So at low concentrations, not all dynamin is inhibited, because remember, that's what map does. So at low concentrations, not much dynamin inhibited. But as the map concentration increases, clearly it's showing you that the proportion's dropping of growth, so there's more inhibition. So it means there's more dynamin inhibition, right? Which means that there must have been more cell death and less cytokinesis. More cell death and less cytokinesis. Got to use every single step from one by one. However, we're not done. We're describing all the phases, if you will, of the graphs. It's nothing, down, 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 nothing, right? Here, then, at these really high concentrations, there was no change. So what's those concentrations between, like, here and here? So here as well, we can see that there's actually no change. It must mean that all of the dynamin must be inhibited. There's no other way to explain it. Otherwise, if there was more to inhibit, it'd keep going down. So then, at the highest concentrations, all the dynamin left must have been inhibited. It's like nothing left to work. That's why there was no further decreases. That's the only explanation for this graph. 0.01 decimeters cubed of map was added to the treated cells. Calculate the increase in the mass of map in micrograms added to the cells to reduce the cell growth from equal to the concentration to 0 of the control. So we need to look at where it's equal, right? So if it's equal to the control, its proportion is going to be 1, and then where it's 0. So here, we're looking from here to where it's 0, which is the differences, if you will. So it's requiring you to be able to read a logarithmic graph. Now, here, 10, 20, 30, right? And then here, 1,000, 2,000. Yeah. That's how you read them. It's just able to fit bigger scales on a graph. That's why it's like that. That's why it looks funky. Because normally your graph is like, I don't know, here, for example, you can see that's going in 0.2s. This is going up by factors of 10. And in between them, they're getting closer and closer and closer. Just to allow us to fit everything on. So here, the numbers we're comparing are those. So the concentrations, so let's just have a look again. The volume that they used was 0.01 decimeters cubed. And they said calculate the increase in concentration. So the difference in the concentration is going to be 2,000 minus 30, right? If we do that, we get 1970, and the unit is micrograms per decimeter, or micrograms dm. Now here, they've told you all of them have the same volume. So in order to make this go higher, they must have changed how much map they dissolve in this, the concentration, which, I mean, told in the question, right? So if that's the case, you can see the units here are micrograms per decimeter cubed. So what that means is that you have 1 decimeter cubed will give you 1970 micrograms, right? So they're asking you how much 0.01 decimeters cubed would result in here. From here to here, what you've done is you've divided by 100. So here we need to do the same, divided by 100. So we end up getting 19.7, which is roughly 19.3 micrograms. And there you go. Look at the units. They tell you what's up. Read it out. Micrograms per decimeter cubed. In 1 decimeter cubed, for every single decimeter cubed, there's this many micrograms of difference in cons. That's all. Okay, so dengue fever is a human disease caused by the dengue virus. Scientists designed an ELISA test to detect antibodies to dengue virus in a patient's blood sample. Figure 10 shows a diagram of this test and some information about how it works. So let's have a look what's going on. So here we've got diffusion of X from X of a anti-human antibody with an enzyme attached. So it's an antibody against human with an enzyme stuck on it. And it's diffusing this, right? We've got C and T, and then we have diffusion from Y, of a patient's blood sample. Fine. So we've got some mysterious thing diffusing from Y this way, and also X, which is an antibody against human, but with an enzyme attached. So there's my antibody, and there's like the enzyme stuck to it, and it's moving in this direction. So what's probably happening is they bind with each other. Now, figure 11 shows the negative and positive results that were produced 20 minutes after the use of, so we have the C line, and then we have a T line. So it's negative if it's only C, and it's positive if you have C, and fine. It says, describe what's on the line at T and explain what causes this line to be positive. So remember that at T, some mysterious thing is happening that's causing it to change. The questions told you that this is an ELISA to detect antibodies to the dengue virus. So just a side note, a lot of people were just saying it detects the virus. That's incorrect. This thing can only detect antibodies that you're making against that virus. So even if you were vaccinated, had a previous infection, or have a current infection, it cannot distinguish between those. All it says is you have antibodies. So the question here says, detect antibodies from the dengue virus. In a blood sample, they must have dengue antibodies, which I'm going to write as this for short, ABS, right? What's happening is that the blood sample and the antibodies are moving this way, and an antibody against this is going this way, right? Something's happening at T for it to do that, for it to just mysteriously make a new line. First of all, if you think about it, if we just run it like this, it's just going to go all the way up here, and then that's it. Nothing's happening. So clearly, the sample has to come up here and stop. So if it's going to stop, these are dengue antibodies. Here, we must have a dengue antigen, right? Otherwise, there's no reason for it to just stop there. That's it. Number two, we wouldn't get a color change there because there's only this dengue antibody that you've made. But if we look, we have another antibody here against the dengue antibody with an enzyme attached. So if the dengue antigen is here, so if I zoom in on that part, let's pretend this is the antigen. For dengue, this is the antibody you made. But now we have this fixed here, and now it's there. So then this other antibody that's going for, this, I'm going to call it X, this antibody here, is now here with a substrate on it. So here, there must be a substrate for the enzyme to react with so that it gives you the line. So in summary, first of all, the antigen needs to be here to actually get your antibody to stick there. Next, this antibody will bind to your antibody there, but it still doesn't give you a color change. It has an enzyme on, so there has to be a substrate there. At that point, there must be an antigen and, the dengue antigen, let's be specific, and substrate. And then what happens is the enzyme on the anti-human antibody binds with the substrate, and you get a color change. That's how they work. So you really need to make sure you're specific with what's going on here. There's no reason for it to just stop. It would just keep diffusing forever. You need to look at what's binding, what's moving this way, what's moving that way, what's binding to what. A lot of people, again, were just saying detection of antibodies or antigens in the blood. Don't do that. Look what the question's telling you. The question can only detect antibodies. At line C, it's shown that the test has worked. So just one reason why a line at C shows that it has worked. Here, if I just rub out all the scribbling I've done now, one sec, right? This is the test when it's not had anything on it. When you run a test, it's negative, you have a line at C. If it's positive, you have a line at C as well. So negative or positive, there's C versus nothing on it, no line at C. So clearly that must mean that it's there for some reason to indicate that, you've done the test, you put your blood on it. So if that's the case, it must mean that the blood has gone up this way and actually moved that way and moved past T, as in the blood's moved, blah, 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 blah. You get a line at C. It must have gone through T, meaning it's worked. It's able to get past that. So that's why it's there. It's to indicate that blood moved through T. Got to remember the context of the question carefully. Figure 12 shows the flowchart of how anti-human antibodies with enzymes attached are produced. So what do? They inject rabbits with anti-human antibodies. They get a blood sample from the rabbit. They fuse B cells from the blood sample with cancer cells. So think about it step by step. They're putting human antibodies into a rabbit. This rabbit's immune system is not going to recognize this antibody. It's going to see that it's a foreign antigen. It's an unfamiliar antigen. It's like, yo, we got to destroy this. Anything the only thing that triggers an immune response is a foreign antigen. So here, the foreign antigen is the antibody itself. So this rabbit's going to mount an immune response. It's going to do the B cells, the clonal selection, the this and the that to make its own antibodies there. So it's got rabbit B cells and they're fusing it with cancer cells. These fused cells that make anti-human antibodies are then cultured and then they get loads and loads and loads of antibodies and then they plonk an enzyme on it here. So it says suggest why the fused cells allow the continuous production of monoclonal antibodies. Look at what a fused cell is. It's the B cells from the rabbit with cancer cells. So for some reason, getting B cells with cancer cells mixed together, as in sticking them together, means you're going to make indefinite, antibody production. Cancer is uncontrolled cell division. So these cancer cells divide by mitosis uncontrollably, right? But that doesn't make the antibodies part. The B cells produce the antibodies. So if you've got these fused cells and they're constantly, making antibodies and dividing uncontrollably by mitosis, they'll be making loads forever. So there you go. That's all it's asking is why each of those components are there. Other people were talking about the humoral response, a lengthy description about it. People were talking about involvement of T cells as well. Why are you doing that? Like, where's that? Come on. Then it says, evaluate the ethics of the production process. So here, you've got to talk about the positive and the negative. You can't just say one thing, right? So a lot of people had missed this. Very few people actually corrected this. A lot of people just gave one side of the argument. When it's evaluate, you need to say both sides, positive but negative. So for example, it's harmful to the rabbits, but, why is it useful? I mean, it, produces useful, medications or drugs, which alleviates human illness. So while, yes, you're causing harm to rabbits, you're also helping people not die. Also, as well as that, you can think, well, I mean, what other problems are there? I mean, you're only injecting them once, you're taking blood, and then you're not using them again. Like, you're not using the rabbits anymore. So it's only, at least it's a one-time thing, or at least you're not killing them, or at least you're just injecting them, but not physically, chopping out their organs or something. The whole point is you got to say the sort of negative ethical problem and then the positive. Well, not positive, but the less harmful evil thing. Early identification of dengue can be difficult as many other diseases make the same symptoms. Early identification is important because people suffering from dengue can become very ill quickly and can need hospital treatment. That is very true. It's not a pleasant disease. Scientists compared the effectiveness of 3 diagnostic tests for dengue fever. So one is a lab test where they take a sample from a doctor's clinic for testing, a new test in the doctor's clinic, and then a new test to be used in the doctor's clinic. So this one's already in the clinic, this one's in the lab, and this is a new one they're fumbling around with. And this is the one that they've described in the previous question. Here's their results. A blood sample from each patient with confirmed dengue fever each time after the onset of symptoms were tested with all 3. So if we look at the table, this is the time after the number of symptoms came on. This is the number of confirmed patients they had tested. So they took, at days 1 to 2, they got 14 patients. Out of those 14, 10 were picked up by the lab test and 6 were from the new test. So that indicates that days 1 to 2, this lab test is better than the new test, and it's actually able to pick it up. The current test was not able to get any diagnosis positive result in those 14, right? So that implies that this is maybe decent, at being used as early as 1 to 2 days after symptoms start. Then, if you look at days 3 to 4, they then took 38 people and tested it. Out of those 38, 28 were from were picked up from the lab test, which is good, suggesting the lab test is the best. But if we look, the new test got 24 of them, which is quite close. So at 3 to 4 days, it looks like that the lab test and the new test are quite close in how effective they are. The current test in the clinic is a bit rubbish, still not very good. If you look at days 5 to 7, they took 18 people. Of those 18, 8 people were picked up by the lab test, implying that the lab test is not as good as these 2 now. The current test is almost as good as the new test, according to these results. So if you look at it, depending on what day it is, one of these tests is better than the other. So at 1 to 2 days, the lab test seems to be the best. 3 to 4 days, the lab test seems to be the best, although the new test is very close. 5 to 7 days, the current test and the new test are the best, and the lab test is rubbish. So just do it step by step like that. The scientists recommended that the new test is used for the identification of dengue fever in all countries across the world. Discuss this recommendation and use all of the information given. So when it says discuss this recommendation using all the information given, what it's telling you is to look at everything and say, is this good? Are they valid for saying this? Is this what in this is saying, yeah, you could do this. So if we have a look, again, I was kind of yapping about it just before. It's better than the current test, but not as good as the lab, except 5 to 7. So 1 to, how many days is that? Sorry, 4, better than current, but lab, right? However, at 5 to 7, was it 5 to 7? It's better than the lab. Let's think about why that would help. If we have a look, they've told you that this new test can be used in the clinic, whereas the other ones have to be sent to a lab. If you can do it straight away in the clinic, this new test might be quicker and cheaper. Because if you look at how it works, remember the previous questions, all you do is you plunk some blood on it and wait to finish its job, whereas you've got to take it to a lab and, wait for things to happen. As well as that, why might they be useful in all places around the world? Well, some countries may have limited numbers of labs as well, or they may have limited training as well. And as well as that, another good thing about it is that days 3 to 4, it's nearly the same effectiveness as the lab based test as well. So it's pretty good. So that's what it's asking you is like, what can you conclude from this data that supports their conclusion and using all of the information, using all the context? A lot of people just kind of said trends and that was it. They didn't say why it would matter in this context. The dengue virus causes damage to capillaries so that blood proteins move out of the capillaries into tissue fluid. Explain how this would affect the return of tissue fluid into the capillaries. So here then, there was a pretty devious misconception. There was a misconception where lots of people said that the return of tissue fluid was by osmosis and not the movement of water. Water is moved by osmosis. Tissue fluid is just simply fluid that surrounds tissues. That's it. The water is doing the osmosis. The tissue fluid's full of all kinds of stuff like solutes, glucose, bits and bobs, nutrients, this and that, blah, blah, blah. Here, work out what's happening step by step. As a quick rundown, arterial end, capillary bed, venal end, back to veins in the heart. What's happening is that because the pressure is so much higher than here, water and small solutes get squeezed out of the capillaries outside of the blood vessels into the tissues. Because most of your blood's flowing through the blood vessels. Why is it now around tissues? That's what's happening. If it does that, then at this end, it's just constantly being shoved out. So it can't actually get back in here. But now if you think about it, water's left, the water potential is going to be low in here. So water comes back in here via osmosis, not the tissue fluid, the water, right? That's tissue fluid. It's just fluid that surrounds it. So here the question's told you that it causes damages to capillaries so that proteins move out into the tissue fluid. Proteins are massive. Proteins don't normally move out. It's just water and tiny things. So for some reason, something's happening that's making proteins get out. The only explanation for that is simply, if we look at it side on, it must have made like massive holes that let it get out, right? So if it's able to now shove proteins out, which normally doesn't happen, then if you think about it, now there's proteins alongside the water in here. So the water potential actually drops out here and it actually gets higher in there. Although, yes, you're losing water, you're also losing proteins. So as a result, you've now got less coming back in via osmosis and more actually wanting to come out and stay out. So that means you have more tissue fluid, less of it's returning. So then that's what we would say is that one is it increases the water potential in the blood, specifically the capillary, and decreases the water potential of the tissue fluid. So then there's less osmosis of water returning, of osmosis causing water returning, I should say, to capillaries. Or on the flip side, you can say more water leaves via osmosis of the capillaries as well. So that's what's happening. So you've got to think logically here. Normally what happens, massive things like proteins and cells don't come out. Here the question's told you proteins can. And they're saying, what could this do to the movement of water back in and out? Fine. So here then, it says, describe how a quaternary protein is formed from its monomers. Do not include the process of translation in your answer. So let's have a look. What were the problems then? So a lot of people got most of the answers just by saying a few lines. You didn't have to yap. The size is, the lines are just there, remember, just to, not have to use extra sheets. A lot of people were just saying that quaternary structure is made of 4 polypeptides. That's incorrect. Quaternary means that there's multiple, or maybe it might have been that they just thought hemoglobin's the only one. That's not true. You can have more than 4, you can have less than 4, there's lots. A lot of people just said tertiary structures instead of many polypeptides, multiple polypeptides, not multiple tertiary structures. And a lot of people said, protein synthesis, even though it literally tells in the question, do not talk about translation. Why? Well, what are you doing? Start from the start. The monomers, they're amino acids. So what do? The amino acids are joined by condensation reactions to form peptide bonds. Like every time you're talking about breaking or joining things together, you need to talk about the bonds and why it's happening. If you do that, I've already secured 2 marks off the bat, right? That forms your primary structure. Next, let's talk about the secondary structure. So what happens is that you've got hydrogen bonds, and that causes coiling or folding. Next, tertiary structure. You have hydrogen bonds, ionic bonds, disulfide bridges between R groups of amino acids, which makes it 3-dimensional. Quaternary structure is where you have multiple polypeptides joined. That is it. You don't have to say anything beyond that. That's all. It's done. Finished. Finito. Describe the structure of DNA and then the structure of a chromosome. So I knew this would have fumbled, to be honest, because everyone gets the words and the bits and the bobs wrong. So let's have a look what's going on here. So lots of people just said phosphodiester bonds and didn't say where they are. Whenever you say bonds, you need to say what they're between. every time. There's no point just saying bonds are here because that's useless. Like there's lots of different bonds and molecules. As well as that, lots of people just said ATCG. Like you have to say what those bases are. You can't just say ATCG, that means nothing. And, start with the fundamentals. Like again, DNA is made up of lots of nucleotides joined together, which is a polymer, a polynucleotide. Do it from the ground up. So if we're talking about DNA, it's a polymer of nucleotides. That's what they are, joined by condensation reactions to form, I'm going to write PDE, but you write the whole thing, phosphodiester bonds. You can't just say where, you need to say between what, So between the deoxyribose of one nucleotide and the phosphate of another, right? So there's that immediately. That's finessed me like 3 marks. We're not done though, because DNA is not just simply one long strand. All we've just said right now is that there's a big long strand. We don't have that, do we? We have a double helix. So we need to talk about the double helix. So what happens is that 2 strands of DNA form hydrogen bonds with each other to form a double helix, right? But we still need to delve a bit more into here, the nucleotides. So the nucleotides, you can't just go ahead and say, you can't just yap and that's it. You need to say why this is happening. We need to describe what they are. So nucleotides, they consist of a deoxyribose sugar, phosphate, and nitrogenous base, which is adenine, thymine, guanine, and cytosine, right? And that's DNA. Now we need to talk about that. That explains everything. That explains how we have this strand. It explains why we have the coiling, and also, the bonds between things. You can't just go ahead and just say like the vague stuff. You got to go from start to finish. Now it says to describe the structure of a chromosome. So in a chromosome, remember, is just a massively coiled, shortened, fattened, condensed helix. Like it's huge. So first of all, in order to make it do that, this DNA is associated with. Associated means it kind of just sticks together, kind of mixed in with there, not really fully chemically properly bonded. And that's what it does. It's associated with histone proteins. And when it coils and condenses, you get chromosome, right? And when do we see chromosomes? For an extra point, you would have 2 chromatids joined at a centromere after DNA being replicated. But I think that's less. I mean, it's a point, but I don't think it's the strongest point. Enough. All of what we've said is enough. There you go. So mutation can result in an increase of genetic variation within a species. Describe and explain other processes that can increase genetic variation. So if you don't know what genetic variation means, and you don't know where you're going from this, genetic variation means you have lots and lots of differences in the gene pool. You have lots of different alleles. Humans are very genetically varied. Some people have black hair, some people have brown hair, some people have blonde hair, some people have ginger hair, different colors of eyes, different heights, different weights, different this, different that, lots of differences. We're genetically very varied. For example, if you look at like a farmer who's farming bananas, they're all identical. There's barely any genetic variation. Genetic variation only exists because they must have had changes in their base sequences. Otherwise, why are they different? And that came from mutations, which must have just been purely random and just led to how we are now. That's entirely what it is. But the questions told you, though, mutations, one of them. So you can't go ahead and just talk about that again. We need to talk about other processes that would make variation higher. So let's see what the problems were first. So a lot of people, for example, were just trying to describe random bits and bobs, and they just could not say what homologous meant. As well as that, the examiner report describes the responses as tortuous, which means full of twists, bends, and crooked stuff. So it's clear that a lot of people were trying to use a model answer or things they just memorized, and it just didn't work. A lot of people just said loads of things and didn't actually say how it leads to increased genetic variation. So, the whole point is we're making new combinations of alleles. We're not making new mutations come in, right? Didn't say that. Lots of people were talking about lengthy answers about natural selection, survive and reproduce, random mating, this and that, blah, blah, blah. Again, that's not what they're asking. We're asking, how do we get more genetic variation, which I said before is lots of more combinations of different alleles. It doesn't matter when you have random mating or, whatever the hell you're talking about. The only things that cause new combinations of alleles, besides obviously mutations, is meiosis and fertilization. It doesn't matter if they randomly mate. It's not the mating that's doing it. It's the random fertilization, and the meiosis that's doing it. All right? Because if you had identical species, if all humans were identical clones with each other, if we had random mating, we're all going to look the same. It don't matter. right? But if we have little bits of variation, we're a little bit different, and we have that random mating, that means we have that fertilization, different mixes, different meiosis, that stuff, that's what's causing changes in the alleles that are different. Now, a lot of people are talking about non-disjunction. That is a chromosomal mutation. That's a problem where you have failure of the chromosomes to separate, which can lead to extra copies being inherited. For example, Down syndrome, which is 3 copies of chromosome 21. And a lot of people were talking about environmental factors causing genetic variation as well. That's not what this topic's about. Loads of people mix this stuff up. When we're talking about inheritance and genetic variation, we're not talking about natural selection. We're not talking about changes in allele frequencies. We're talking about the actual alleles being different in you. If your mum has black hair and your dad has blonde hair, you're probably going to have black hair because you have a different combination of alleles, but you'll have combinations of your mum and dad. So that's what it's trying to get at. It's nothing to do with changes in allele frequency. Changes in allele frequency means more people over time would have blonde hair, more people would have black hair. Just like, look, you got to be careful with what you're saying. You can't just yap. What processes increase the different variation combinations? The key ones are random fertilization. You won't have, you can't predict which sperm is going to get to which egg. So immediately, that's one of them, right? Of the gametes. Because every single sperm you have will be different, and every single egg you have will be different. As well as that, I need to think about why they are different. So one thing you can have is you have independent segregation of homologous chromosomes. I'm going to elaborate on what that means, right? And then we also have crossing over between homologous chromosomes, right? But then you need to say what the consequences that this produces new combinations of alleles. Nothing to do with frequencies. So if we look in a bit more detail, yes, random fertilization is great, but we need to talk about these other processes that lead to the random fertilization actually having their effect. Number one thing I see people fumble horrendously every single time is what a homologous chromosome is. A homologous thing is things that are like representative of each other, two different things that represent the same thing though. So for example, here's me, miserable after reading some of the states of these answers. I'm joking. And here's you. It's not your guys' fault, don't worry. You just guys get taught like absolute garbage sometimes. It's fine. Right? This is my right hand. That's your right hand. They're homologous. They're two different right hands, but they are both right hands. So they are homologous right hands. So homologous chromosomes then are pairs of matching chromosomes, right? So it's one maternal, one from mum, and one paternal. So humans are diploid. We have 23 pairs of chromosomes, 46 in total. So if I look at chromosome 1, it's not just like one there. We've got one there as well. So we have maternal and paternal. Those are homologous chromosomes because they're the exact same genes. It's just there's different alleles on them. So for example, if your dad is blonde and your mum is got black hair, then they still both have hair color. It's just the alleles are different, right? There's something to really, really understand. When you're doing mitosis, the homologous pairs do not separate because if you separate them, you only have maternal and paternal in separate things. You become haploid. We are diploid, so we have two pairs. They're also not bonded together right now in a normal state with a centromere in the middle. This is another misconception. As a side note, going on from here, let's talk about meiosis. So I'm going to pretend that we have some mystery organism that has one singular chromosome in it. All right. So maternal is blue, paternal is purple. So it's a diploid organism with a maternal and paternal chilling in it. There you go. And there you go. So remember, these are homologous chromosomes. It's diploid, so its N number is 2N. When you have meiosis, number one is we replicate all of the DNA. Otherwise, you can't split this any further. So when we do that, once we've replicated the DNA, we end up with 2 times 2N, double the amount. If you put 4N, that implies that you've got 4 homologous pairs and like 4 different parents, as in a tetraploid, which you're not. So what happens is now you have this here and here. You've got 2 sister chromatids bound by a centromere. Another common mistake people make is not realizing that these 2 sister chromatids are copies of the exact same chromosome, as in this is maternal. It is not this. You are not mixing together paternal and maternal like that. That's incorrect. That's not, why is that? That's not happening. This is DNA replication making that. It's the same for mitosis. When you are replicating them, you are simply just copying the same thing. So again, maternal, paternal, number 1, right? In meiosis 1, you need to understand what the whole purpose of it is. Meiosis 1, all that happens is your homologous chromosomes separate. But before they do that, what happens is, well, they separate. What happens is you get crossing over first, and then they separate via independent segregation. What that means is you can't predict which cell this is going to end up being. So if you look at crossing over real quick, again, to reiterate, maternal, centromere, paternal, right? Crossing over is where you have these chiasmata here, and what they do, chiasmata is where they cross over, and you end up making new combinations of different alleles. So for example, here, I might have a bit of maternal here chilling, right? And then I might have crossed over this part here. So now could you see there's a mixture of paternal here? And then for paternal, the paternal geezer here, it'd be this, and then that. Still paternal. That's what's happening. These homologous chromosomes are crossing over and exchanging alleles to make new combinations of alleles. As a result, you have new bits and bobs coming in. So for example, let's pretend I do cross over 2 bits, right? So I'm going to end up having here and here, right? Let's pretend this region so this region is blue eyes paternally and then brown eyes maternally, right? And then if we look here, let's say, for example, this is maternally, she's tall, paternally, he's short. Cool? So if we look at the original maternal, remember, maternal was tall and brown-eyed. If we cross over these alleles, now we're getting a whole mixture of stuff going on. Because if we look, can you see how paternal, blue-eyed, remember, this is now giving you blue eyes, and this is short. Can you see how that one's not really changed? So that one's going to be brown eyes and tall. Whereas here, now we've exchanged that. That's going to be brown eyes, tall, and then that's going to be the same as paternals, which was just, what was it again? Blue eye. Can you see how we got new combinations out of that? That's what's happening, is crossing over is making new combos of alleles. So when those homologous pairs separate, you end up with 2 daughter cells that now only have either maternal or paternal, but you can't predict which one ends up with what. As a result, this is at the point where you become haploid. So you have 2 times N, because you've only got maternal here and paternal. 2N means maternal and paternal are there. Now, if I use this example here, we now have this chilling here with a bit of purple stuck on the end here like that. And then we have this chilling here with a bit of blue sticking on the end like that. So can you see how we have new combos compared to the original? If we didn't do this, we just end up with the same thing next step. Next, what happens in meiosis 2 is, well, we've separated the homologous chromosomes, we need to now separate these. So what happens is the sister chromatids separate now. This is also by independent segregation. So what ends up happening is you get 4 daughter cells that are genetically different, because now what happens is, remember, they're haploid, so they only have 1 copy. So this splits, remember that 1 maternal from original? We've now got this 1 maternal with a bit of funky stuff going on there. And then we have this 1 maternal with a bit of funky stuff on it too here. And then we've got the original paternal. So if we look at the characteristics I gave them, remember, this gamete maternally was, brown-eyed and tall, right? And then paternally, what ended up happening? We have brown eye, short, right? And then maternally on this side, we ended up with blue eyes and tall. Whoops. And then here, we have blue eye and short. Can you see how we have lots of different combos now of things that never existed to begin with? Because mum was brown-eyed and tall, dad was blue-eyed and short. And now through crossing over, recombination, all of this blah, 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 we now end up with new combos like that. So to summarize, just as a quick thing, because I know that was a lot of yapping, homologous things are 2 matching things that represent the same. So my right hand, your right hand, different hands, different people, but they're still right hands, so they're homologous. Matching chromosomes, it can be pairs, it can be triplets, it can be quartets, however many, depending on the organism. They are matching chromosomes, right? In a normal cell, you're chilling with 2N, maternal, paternal. Before mitosis and meiosis, we have to replicate everything, so we double these all, right? So you end up with 2 lots of maternal, paternal. You do not mix the 2. The first step in meiosis is you want to separate the homologous pairs. So maternal goes one cell, paternal goes another. But before that also happens, we have crossing over. Crossing over is where you get a new mixture of these things, right? So maternal, remember, was just brown-eyed and tall. Paternal was blue-eyed and short. Now we end up with brown-eyed and tall on this chromatid, and whatever the other mixture was, right? And that's the process here. That makes new combinations of alleles. Immediately, one way of making genetic variation higher, because the kids won't match the parent. Then what happens? You get independent segregation again, which randomly assorts them into these, and also here as well to further do it. So you're not going to end up with the same gametes made every single time. And as well as that, you end up with 4 genetically different gametes like that. That explains why you're not identical to your siblings. Because if these didn't happen, yes, your parents are different, but then you would be literally all the same. But because of this, that's what makes you different. And that's why random fertilization of gametes lets that happen. So it's really important to understand this concept and not mix and match words and not memorize things either, please, for the love of God. So again, genetic variation, lots of different combinations of alleles, some tall, some short, some this and some that, right? And processes that lead to that is, well, the random fertilization of gametes is indeed one, but that doesn't explain it all, because what if the gametes are all identical? It's these processes that make it different, and mutations as well. And if you look, remember, mum was brown-eyed and tall, dad was blue-eyed and short. Now we have a kid that could end up being brown-eyed and tall, a brown-eyed and short, a blue-eyed and tall, a blue-eyed and short. There's so many more combos that never existed to start with because of that. We've got these new combos coming in. Okay, so that wraps up the paper. Please like, share, and subscribe. You know the drill. If you guys have any questions, drop them there. If you spot any mistakes, let me know. And also, if you have any feedback for future sessions or anything you want me to make, like topic videos, let me know. I also do want to start making more content, like, about studying and how to do things, how I do things, this and that, what life is like, little funsy stuff like that. But I hope that helps. Thank you so much. Bye bye.